everyone. My name is Aleksandra Nivina. I'm a second-year PhD student here in the CRI, and I'm doing my thesis in synthetic biology. So today I'm going to present you the synthetic biology in general, and I will also talk about the interactions between synthetic biology and design from the synthetic biologist's perspective. So first, I have to tell you that there is no definition of synthetic biology, no unified definition of synthetic biology. So every source gives its own definition with a little bit of difference. But what I try to do is to sum up all of those definitions. So I copied all the definitions and feed it to the Wordle, which is a word cloud generator. And so here the size of the word basically corresponds to the frequency at which the word occurs in the definitions. So as you can see, synthetic biology can be defined through biological systems, which is logical. This is what synthetic biology deals with. But if you look at other words, you can see words like parts and devices. And you can see these words because synthetic biology stems from an engineering background, and so these kinds of terminological words like parts, devices, systems, they occur in synthetic biology as well. And if you look at other words that we see, those are engineering, construction, design. Here we have redesign. So you see that design is really important in synthetic biology, and a lot of people define synthetic biology through design. And if you would like still to have like some kind of definition, I'd say that synthetic biology is a discipline that engineers or designs biological systems from existing parts and devices, or re-engineers existing systems that we can find in nature. But what's more interesting is actually not how synthetic biology is defined, but what it could do. So I will briefly explain to you some of the applications. Some of them are real, some of them are in development, and some of them are just our imagination of how the future could look like. For instance, synthetic biologists are already capable of engineering bacteria that can produce fuel from sugar. Unfortunately, this production of fuel is not economically viable for now, because fuel that we can extract is less expensive, but it could be still a possibility for future applications. Another possibility for synthetic biology applications is to engineer bacteria or yeasts or other organisms that could produce drugs. For instance, a yeast and the bacteria have been engineered to produce uracemic acid, which is the precursor of a drug that's used to treat malaria. But other drugs are also possible applications. Everything is open for future developments. There are also some applications for cancer treatment, to design cells to treat or to detect cancer. But these are still in development. There's also a possibility to design new materials with synthetic biology. For example, there are bacteria engineered that are capable to produce monomers of spider silk. So you might know spider silk as it's one of the most one of the very strongest materials that we know of, and it's natural, and so the bacteria are capable of producing monomers. They still cannot make a thread of it, but maybe in future we will find a way to thread this kind of threads of spider silk. But you can also imagine other new materials, for instance, to use in our bodies or outside of our bodies. Another application is in gene therapy. You could imagine some technological advances that could allow better delivery of gene therapy in cells and stuff like that. Also, another application is in bioremediation. Bioremediation is basically a detoxifying environment from things like oil, petroleum, plastic, radioactivity, and other toxic chemicals. And the bacteria are already capable of purifying the environment from these molecules, but we can engineer bacteria to even better degrade those toxic chemicals. Bacteria are also very good at detecting molecules in the environment and responding in some fashion. And so an interesting possibility for synthetic biology applications is developing biosensors. We can imagine sensing toxic chemicals in the environment, or maybe some disease markers in the blood. And we can use these in some kind of devices. Basically put bacteria inside, and then they produce their function, basically of identifying some molecules and then reporting to us in some way, for instance, with a color, or with light, or in another way. Another interesting thing is that in our bodies there are a lot of bacteria living both on our skin and in our gut, and they're very important for our life. And we can imagine maybe modifying this microflora. There are both bacteria and some other microorganisms living there. And we know that this microflora might be responsible for a lot of diseases. It might be related to the emergence of a lot of diseases. And we could imagine that maybe by reprogramming this microflora, we could possibly make ours healthier, or resist diseases, or treat diseases. But this is just future applications. For now, it's not. There's no real application for this yet. However, there's a lot of progress being done on engineering crops, because you can do synthetic biology in all kinds of organisms, in bacteria, in yeast, in mammalian cells, and also in plants. And you can imagine improving crops by increasing their productivity, or increasing their flavor, or making them resistant to some viruses, etc. And this could also be a solution for feeding the population. Another application, maybe that would not save the world, but still it's very profitable, is making cosmetics. There's actually a company that extracts some oil from algae, and they put them into creams that apparently prevent people from aging. And so you can thus think about synthetic biology application in treating people from aging and preventing that from it. So I've mentioned several of these applications, and actually, as you can see, you can find the same 5 notions that we're going to be talking about in this expedition. So identity, because the gene therapy and the microflora are tightly connected to your identity. Drugs and therapies could influence your health. Of course, cosmetics and anti-aging products would have an impact on beauty. New materials and maybe new implantable things could enhance our performance. And of course, biosensors, they would improve our ways to sense the environment and to analyze it. And so now I'd like to talk more about the connection between synthetic biology and design. So you might know that design actually has can have different connotations. There are several senses to the word design, and synthetic biologists not necessarily use it in the same sense than classical designers. Because when we use the word design, we usually use it in phrases like designing a genetic circuit, or creating proteins by rational design, or designing an experiment or DNA fragment. So what we mean when we use the word design is that we want to create something with a purpose, we want it to accomplish its function, we want it to be rational and very thought of. I'd like to present you briefly what is, for instance, design of a genetic circuit for a synthetic biologist, because this is the very basis of all synthetic biology applications. It's what we do on a gene level. So, for instance, imagine that you would like to produce some useful proteins, some useful proteins B and C, in response to a molecule A that circulates in the environment, or maybe you want it to act as an inducer, basically to trigger the response of the cell, and it will produce proteins B and C. So I guess a lot of you are accustomed to seeing code. So actually, engineering cells can be very similar to programming on a computer. So this phrase, producing proteins B and C in response to molecule A, can be written in this way. So if the concentration of A is higher than a certain threshold, then you produce B, but you produce C, and you end the execution of this function. And you can see the biological, like the genetic coding in the same way. So you have, this is DNA, and you can have a sequence of DNA called promoters that react to directly or indirectly, for example, to a concentration of a molecule in the environment. So in response to a certain concentration of molecule A, downstream of the promoter will flow the gene B and the gene C. These are the functions that we want to execute, and in the end, we put a transcriptional terminator to say that the function that we want to execute is finished. And this is how we code the gene circuit. And basically, assembling these parts one by another is what we mean when we say we design a gene circuit. So we select which part we're going to use and why, etc. So this is a very, I'd say, simple genetic circuits, even though they weren't so simple 20 years ago, or even 10 years ago, because just the first circuits of this type appeared around the year 2000-2001. But now we are already capable of engineering more complex systems. So instead of just putting 2 or 3 genes, we can basically tackle with a metabolic pathway in an organism, because an organism makes several chemical reactions, and they're all interconnected with each other. And for example, we can enhance some reactions and stop other ones to divert the chemical potential of the cell towards producing the molecules that we would like. And so this is how, for example, people are capable of making bacteria produce fuel, or produce some plastic, or produce some drugs. And now we can even think about designing organisms, because first what's called synthetic organisms have been produced. By meaning synthetic, we mean that the DNA has been synthesized and then inserted in a living cell. But still, this means that we can code the genome and then insert it in a living cell, and the cell will produce the functions according to what we have put inside of this genome. So this is how, this is really what we understand by the word design. In synthetic biology, when we design systems, we want them to have particular characteristics, such as being simple, being robust, being efficient, biosafe, etc. And actually, if you want, if you could look at other areas of design, you can find some similarities. For example, if you think about urban design, instead of saying simple, you could think about a building, or a community, or a certain appliance in case of product design, easy to use. Instead of saying robust, you can think about sustainability, about things, objects, design being practical. In synthetic biology, we're very concerned with biosafety, because there are very strict regulations that say what you can and you cannot release into nature, because genetically engineered organisms might might do some harm. So biosafety is a big issue for synthetic biology. And now eco-friendly is also a very important issue in other areas of design. Also, both in synthetic biology and other areas of design, we have to work under some imposed constraints. For instance, in synthetic biology, there's a big issue with part availability because we only can work with genes that we already know of and we know how they work, and it's a very limited amount. And also, they're not always compatible, so you cannot put any two kinds of genes together. There might be some crosstalk and some other problems. And also in other areas of design, you might have some constraints with material availability, non-compatibility with some techniques or some materials that you're using. In synthetic biology, we're concerned about ethical constraints, and in other area designs, there are always some guidelines and regulations that might also say what you can and you cannot do. So actually, if you look at that, you can see that maybe synthetic biology really isn't that different from other areas of design. And you can think that we can actually consider synthetic biology as a new area of design. Oh yes, and of course, both of these areas have a lot of creativity, because when you design a biological system, you have to be creative. You want to decide what you want to do and how you want to do this. And the same goes for design, of course. And so if you regard synthetic biology as a new area of design, there are actually a lot of different ways for scientists and designers and artists to collaborate. There can be just one-to-one collaborations, as in case of a group called Synthetic Aesthetics. There are pairs of artists and scientists who work together on interesting projects. You could also think about applying design to synthetic biology for some product development, or also for finding new applications. One of the very well-known examples of applying this kind of biology to maybe synthetic biology to the creation of new products is one artist that decided to produce clothes and shoes from kombucha, which is a symbiotic growth of fungi and bacteria. So you can find new applications for already existing things. Design can also be applied for developing computer-aided design tools. For instance, Autodesk is involved in developing this kind of tools for synthetic biology. You can think about design in terms of process design, and you can also, of course, collaborate between science and design for creating art. This, for instance, is an art piece created by Petri dishes, and when you see darker pixels, it means that there are bacteria growing on the Petri dish. So as you can see, synthetic biology can be regarded as an area of design, to my opinion, and I believe that a lot of collaborations can be made at this frontier, and I believe that we can all speak the same language and find the ways to share our concepts and our beliefs. So thank you very much, and I hope you'll have a nice exhibition.